fortunate to have Dr. Florian Krammer, and I have the honor and privilege to introduce him to you all. Dr. Krammer has earned his PhD from the University of Natural Resources and Life Sciences in Vienna, Austria in 2010. He joined Mount Sinai for postdoctoral training, working on hemagglutinin stock-based immunity and universal influenza virus vaccines. Since 2014, he's been an independent investigator studying interactions between antibodies and viral surface glycoproteins and the translation of these findings into therapies and vaccines. He serves as a professor of vaccinology within the Department of Microbiology, and in addition to authoring over 100 peer-reviewed articles, is a member of the editorial boards for the Journal of Virology, PLOS ONE, and Helion. A very warm welcome to Dr. Florian Krammer.
Thank you very much for this nice introduction, Dr. Mustafa. I'm trying to share my screen here. That should work. All right. So I hope you can all see my screen. What I'm going to do today is I'm going to talk about two aspects that are related. One is immunity to SARS coronavirus 2, and the other one is vaccines for SARS coronavirus 2. And as you've seen in the last few weeks, there's a lot of excitement about them, and there's a lot of new data. Just here, my disclosures. I think the only thing that I really should flag here is that my laboratory is collaborating with Pfizer on animal models for SARS-CoV-2, not necessarily vaccines, but animal models, and that's certainly relevant for vaccine development. So I want to start out with this slide about the structure of SARS coronavirus 2 and its surface glycoprotein. By now, you're probably very familiar with the structure. I have a schematic here on the left side. We have the RNA genome on the inside. It's covered by nucleoprotein. There's a lipid envelope that has two envelope proteins, actually the E or envelope protein, which is an ion channel, and the matrix protein. And then we have the spike protein on the surface. And the spike protein is really very important because that protein is what the virus uses to interact with our cells and what it uses to bind to the human receptor. And so on January 10th, the sequence of the new virus was released by colleagues at Fudan University. And we downloaded the sequence and started to build tools and reagents to measure immune responses, antibody responses to the virus. That's something my laboratory does a lot for influenza. And so that was, of course, the first thing we were interested in. So we started to make recombinant versions of this spike protein, which is also shown here on the right side. This is a crystal structure of this trimeric, homotrimeric protein. One monomer is shown here in dark blue. And the receptor binding domain, so really that part that interacts with human ACE2 is shown here in red. So we started to make these proteins recombinantly, and we started to set up serological assays to measure antibodies. Why did we pick this protein and not, for example, the nuclear protein? The reason was that it's known for coronaviruses that antibodies to this spike protein can neutralize the virus, which makes sense because if something big binds here, the virus can't interact with our cells anymore. And so that was the rationale for using that target. And so we started pretty early on. We had an assay up and running, simple ELISA to measure antibodies. You can use that for measuring IgG, IgM, IgA. We had that up and running in February, and in March we started to talk to our colleagues in pathology, Dr. Carlos Cordon-Cardo, and they were interested in setting this assay up in the CLIA lab so that they could start to screen samples for antibodies and identify seroconverters that may then be recruited as plasma donors for plasma therapy. And so we set this up initially as a two-step ELISA where we screen at one serum dilution against the receptor binding domain, which we made recombinantly. And if that signal was above a cutoff, we performed the second ELISA where we quantify then antibodies binding to the full-length spike. And this sequential ELISA setup gives us a very high specificity, and the sensitivity was also reasonably high. This got New York State emergency use authorization still in March, and then an FDA emergency use authorization in April. And a lot of the data that I'm going to show you is based on this assay. This slide is probably outdated, but Mount Sinai has screened more than 70,000 donors, potential plasma donors by ELISA. I think it's 80,000 now. Those are mostly mild cases, and they come in two categories. Initially, we had PCR-confirmed donors, so people who we knew had the infection, and donors with suspected COVID-19, but without a PCR confirmation. So far, more than 500 patients were treated at Sinai with convalescent plasma. When we got the initial about 1,300, 1,400 samples, we did an analysis and we analyzed how many of the PCR-confirmed donors seroconvert in our assay and how many of the suspected donors seroconvert. And we saw that of the PCR-confirmed donors, 99.5% seroconverted, meaning that a lot of the people, or the vast majority of people who had a confirmed infection made an antibody. For the suspected donors, especially in the beginning, it was only 40%, and that's probably because if you go back to March, February, there was also influenza around, there were other respiratory viruses around, and people who thought they had COVID-19 might not have had COVID-19, but a different infection. And together with our colleagues in pathology and together with Anja Vanberg, we started to track this. And of the 70,000 people screened, we found that about 30,000 were positive. And so the clinical lab has these titer categories, you're probably familiar with them, where they have low titers, which are 1 to 80 and 1 to 160. And of the 30,000 people, only 7% had these low titers. Then we have medium titers, which are classified as 1 to 320. And about 20% of individuals had medium titers. And then we have 1 to 960 and 1 to 2880 or higher, which are considered high titers, and about 70% of individuals have these high titers. And if you look at screening over time, so this is just positives per day, and the blue line is showing you positive individuals with a titer of 1 to 320 or higher per day. And this oscillates around 90%, meaning basically that people who get infected make usually a relatively robust antibody response, which I think is good news. We also wanted to see how functional these antibodies are. You know, ELISA is a binding assay. Binding antibody doesn't necessarily tell you much about neutralization. So we also performed neutralization assays. This is done in biosafety level 3 in a microneutralization assay format with authentic SARS coronavirus 2. And there we measure an ID50, a neutralizing dilution 50. And when we plot this against the ELISA titers, we see that there's a very nice correlation. The higher your ELISA titer, the higher your neutralization titer. And specifically, if you're at 1 to 320 or higher, almost everybody has neutralizing activity in their serum. So that was great. The next question that came up, and you probably remember that in spring when there was a media hype about waning antibodies that would go away within weeks. And so that was our next question. And this initial media reporting about waning antibodies was actually triggered by a paper in Nature Medicine. And I always bring that as example for bad science communication. So what they showed in this paper was that they measured antibodies in asymptomatic and symptomatic cases in initial measurement, and then 8 weeks later. This was done with a non-quantitative NB assay. And what they saw was that in the asymptomatic cases, in about 40% of individuals, they lost antibody binding completely over 8 weeks. It wasn't as bad in the symptomatic cases, but this was reported, and then there was a lot of doubt about long-lasting immunity. So we started to look at that too. And so we had 121 plasma donors that gave a sample on day 30. They were recalled around day 82, and then at day 148. So that's about 5 months out. And what we saw there was that the titers initially are relatively stable. So between day 30 and day 82, there was not much movement in terms of the titer. So GMT is here geometric mean titer. And then there was a drop-off between day 82 and day 148, and that's what you would expect if you look at B-cell biology. We also stratified that by initial titer, and we see, yes, if you have an initial high titer, you see this drop-off. But it was interesting to see that if you have a lower titer, you get initially even an increase between day 30 and day 82. And then it goes back to the titer levels of day 30 at 5 months out. And this is interesting. We see this a little bit delayed antibody kinetics in mild cases quite a bit. And so this actually goes very well with another panel that was in the same figure of this Nature Medicine paper, where they actually looked at neutralizing activity of those sera, where the asymptomatics were actually higher and certainly didn't lose neutralizing activity over 8 weeks. And the same is true for the symptomatic cases. So it always depends on what the popular press really picks up on. And basically, even in that paper, they showed that neutralizing antibodies were relatively persistent, and that's what we measure also with our ELISA, antibodies to the spike protein that are neutralizing. We also looked at neutralizing activity over time in a subset of our 120 plasma donors, and you see basically the same. You do have a drop-off in geometric mean titer. This is now neutralizing activity from about 180 to 80 and then to 52. But certainly none of these titers just disappear. We also see that there is still a very nice correlation at 5 months out between neutralizing activity and ELISA endpoint titers. I don't know what's appearing on my screen here right now. I don't know if anybody sees that, but there's some kind of, I'm just going to ignore it for now. And so what does all of this mean, right? Does this mean we make these antibodies, we are protected? And that's a big question. And so initially, in spring, Dan Baruch's group did 2 very elegant, very nice experiments in non-human primates, in rhesus macaques, which are a good model for SARS coronavirus 2. And they were reported back to back in Science. And so here on the left side, you see an experiment where they infected these rhesus macaques with SARS coronavirus 2. So these are basically RNA or titers in the bulb. And you see that the virus replicates very well out to day 21. And then a few weeks later, they re-challenged these animals. Basically, they tried to infect them again, and you can see that this was not successful. We have very little RNA that can be detected in the lower respiratory tract here. It's basically probably input virus in these 2 animals, and all other animals didn't even have a signal. So infection can certainly protect from reinfection in this animal model. In addition to that, they also did an experiment with a vaccine, a relatively imperfect experimental DNA vaccine that gave a variety of different neutralizing antibody titers. And when they challenged the animals and looked at the copy numbers of virus genome in the lung, in the bowel, and they correlated that with neutralizing antibody responses, they saw that neutralizing antibodies basically correlated with protection. And if you look at the titers here, the titers are actually not that high. This is a 1 to 100 titer. So what they found, at least in this monkey model, was a correlate of protection that was neutralizing antibodies. And this is not something really new. We have correlates of protection for many vaccines and many diseases. Here are just some examples: hepatitis A, hepatitis B. For influenza, we have this famous 1 to 40 HI titer that gives you 50% protection. And then certainly, for example, for measles, we have microneutralization that also correlates with protection if you have a micronewt titer of 1 to 120. And so we need something like that also for SARS coronavirus 2 and for SARS coronavirus 2 vaccines. And there are already first insights into that from two studies that I wanted to show you. The first one was published a few months ago and actually got a lot of media attention. There was a New York Times article about it. It's from the Krammer lab in Seattle. And so they enrolled the crew of a commercial fishing vessel. 122 individuals were on the boat. They bled everybody. They measured neutralizing antibodies, and only 3 of them had neutralizing antibodies. And then the fishing vessel left to go fishing on the Pacific. And as it happens on these ships, sometimes there was an outbreak, and there is very close quarters, people interact with each other, and they had an attack rate of 82.5%. Interestingly, the 3 individuals with neutralizing antibodies were not infected. They were protected. And these are small numbers, but they are statistically significant. And that was a first hint at a correlate of protection. More recently, there was a preprint from a British group. They monitored healthcare workers and also found that antibody titer correlates with protection. They show this in this graph on the right side here. It's graphed a little bit weird, but basically what you have on the x-axis here is the antibody titer against the SARS coronavirus 2 spike. 8, this line here at 8 marks what's considered positive. So everything on the right is positive, and everything on the left is a negative. And then they show this here as incidence of PCR positive results per 10,000 healthcare workers. It's a weird way of showing it, but basically what they're saying is, yeah, if you have antibodies, your risk of getting infected, and these are asymptomatic infections, is much lower, and the more antibody you have, the lower your risk is. And so together with my colleague Viviana Simon here at the Department of Microbiology, we also wanted to start a study like that, and we did back in April, and the study is called BERRIS, or Protection Associated with Rapid Immunity to SARS coronavirus 2. And the aim here was to investigate if reinfection is possible, how frequent it is, and to determine a protective titer. And so we enrolled 400 individuals, now I think a little bit more than 400. About half of them are already antibody positive, and about half of them are antibody negative. And we collect serum and PBMCs from them basically every 2 weeks. We perform serology. Saliva is also collected so that we can go back and test if somebody was positive for the virus, if you see a blip in serology, an unexplained increase. And we're also looking at samples if somebody becomes symptomatic with a biofire respiratory panel to exclude other viral infections. And so we're doing this since April, but of course, as you know, the attack rates in New York City were relatively low until recently, and so we didn't have a single infection in the whole cohort. What we do have are antibody titers, and they're basically telling us the same story that we have seen with the other longitudinal samples. And that's what I'm showing you here. Again, we have antibody titers here. They're now shown as area under the curve. Day 0 here is not really the day of infection, it's really the day of enrollment. Many of the people we enrolled actually were infected a few months before they were enrolled. And so what you can see here is basically the same. We have some drops in the beginning, and then the titers stabilize over time, which confirms the data that we have from the other study. Again, because we have only 400 people in this study, and we had very low attack rates. We were asked by NIH to expand that study, and it's now taking place at 9 sites across the US with a target enrollment of more than 10,000 individuals. Right now, I think about 6,000, 7,000 are enrolled. So in conclusion, for the antibody responses, we see that humans induce solid antibody responses to SARS-CoV-2 even after mild infection. The antibodies that bind to the spike protein correlate with neutralization. and the antibody response looks pretty normal. We get an initial strong increase, which is driven by plasmablasts. They secrete a lot of antibody, but these cells die relatively quickly. The antibody, the IgG, has a half-life of 21 days, so it stays stable for some time. And then it starts to decline a little bit and basically stabilizes then at a certain lower level, which is driven by long-lived plasma cells in the bone marrow. Of course, the $1 million question now is that level in an individual is above or below the protective threshold. The Berry study is ongoing and was extended to other locations across the US. And what I didn't show here, but we are taking part in some of these studies, we also get a relatively solid T cell response, which is mostly focused on CD4 T cells and also memory cell responses for both T cells and B cells after natural infection. We're doing some work with Shane Crotty and Alex Setti at La Jolla Institute of Immunology for that. And now I wanted to switch gears to vaccine development. And I wanted to start out with a schematic of how vaccine development usually works, and then I'll show you what happened, what was changed for SARS coronavirus 2. So at the microbiology department, we have a few vaccine development projects, and believe me, most of them are going very slow. It takes a very long time until vaccines move into the clinical phase, and then the clinical phase is also a very long journey. We have a lot of experience with that, and sometimes it's really a glacial pace. Typically, vaccine development starts with design and exploratory preclinical experiment. You play around with a vaccine concept in the laboratory, you try to vaccinate mice, you try to protect them, and that might take years. And once you have a concept and antigens that you think are working well, you typically need to look for a commercial partner, because then you start process development, GMP process development, preclinical, more formal preclinical studies, toxicology studies, and that's usually not really what an academic lab can do. And this phase usually takes about 2 to 4 years. Once you have all the data, you submit an IND and you start a phase 1. And then in the phase 1, you basically look at safety and to some extent immunogenicity in a small number of subjects, usually below 100. This might take 1 to 2 years, and depending on the results, you might actually go to phase 2. But you really have to think hard, because these things are very, very expensive. If you go to phase 2, usually what you do there is you look again at immunogenicity, you look at different dosing regimens, different intervals for prime boost vaccinations. This might take another 2 years. And then you make hard decisions, because after phase 2 results, even if they look good, there's a lot at risk for phase 3, because phase 3 studies are extremely expensive. You need to reevaluate if there's still a market. You need to reevaluate how well the vaccine works, what chances it has to make it through phase 3. And so a lot of vaccine development drops off after phase 2. And then you do the phase 3 studies, which are usually also taking 2 to 3 years. And if you meet the predefined endpoints, you might submit the biologics license application to the FDA or the EMA in Europe. And they might still take time until they license the vaccine. And so the whole process is about 15 years. And after that, a vaccine company starts to produce and distribute vaccines. They wouldn't start before that. That would be economically problematic. Now, for SARS-CoV-2, this was basically crunched to 10 months to 1.5 years. So we probably have a vaccine on the market by the end of this month. So the development took a little bit less than a year. And this was possible because, first of all, money was not really an issue. There was a lot of budget available, and that, of course, helps to shorten timelines quite a bit. But we had also other advantages. First, the design and exploratory preclinical phase could be left out because there was already a lot of existing data for SARS-CoV-1 and MERS coronavirus. So basically, once the sequence was available, people just designed their initial vaccines, and those went into phase 1 trials. The first one actually started already on March 16th. Processes were adapted from other vaccine production processes, so that was not really an issue. And a lot of the development here was done in parallel while phase 1 trials were already ongoing. A lot of phase 1 trials were actually adaptive phase 1/2 trials. And when interim results were available, phase 3 trials were already started. And at the same point, vaccine producers started to produce vaccine batches at risk. And now, I mean, as you know from the media, the Pfizer and Moderna already applied for an emergency use authorization, which is different than a BLA. Emergency use authorization also takes the whole situation into account. And so that shortens the process as well. And so therefore, it's likely that we have vaccines available within a year. What is important is that there were no shortcuts in terms of safety. The safety data still needed to be there, and the shortcuts were really mostly because of the financial situation and basically unlimited funding. I just wanted to give an overview of different vaccine candidates that are out there. Here in the middle, you again see the schematic of the virus and the spike protein. And so there are many different ways to make vaccines. One is to just grow the virus and then inactivate it. This is what's shown here. And those are vaccines that are right now developed in China and in India. There's actually 3 of them in phase 3 trials. You can also attenuate the virus either by passaging it under non-ideal conditions and wait until it adapts to that conditions but doesn't grow well in humans anymore, or you can genetically modify it, take away maybe some of the open reading frames. This can be done for SARS coronavirus too, but none of these vaccines is yet in clinical trials. So it's a little bit more complicated and takes time. Then we have vectored vaccines. They can be replication competent. That's usually done with a vaccine virus or non-replication competent. The ones that we right now see are basically adenoviruses. And what you do there is you take a virus, if it's a vaccine virus, you manipulate the virus and put in the sequence for the SARS coronavirus 2 spike protein so that when you vaccinate with that virus, it also expresses that protein. Or in the case of adenoviruses, you basically remove parts of the genome. The virus can't replicate anymore. You clone in the sequence for the SARS coronavirus 2 spike protein. And when you use that as vaccine, the virus doesn't replicate in your cells anymore, but it expresses the protein. And this is what Janssen and AstraZeneca are doing. Then we have different forms of recombinant protein vaccines, virus-like particles, just the receptor binding domain or the spike protein. And specifically the spike protein vaccines are now in phase 3 as well. There's one produced by Novavax that looks good. We also have DNA vaccines, which went into phase 1 quickly, but there are holdups now. They haven't been moved forward that quickly. You can also make virus vectors that display the spike on the surface, and they are basically treated similar as inactivated vaccines. And our department is doing that with Newcastle disease virus. So Peter Belezi's lab, also in collaboration with Adolfo Garcia Sastre and me, has a large project ongoing with these types of vaccines. And then, of course, we have the RNA vaccines, where you chemically or biochemically synthesize a messenger RNA that codes for the spike protein, and you package that into lipid nanoparticles. And when you inject that, this is taken up by muscle cells mostly. They express the spike protein, and you get an immune response against the spike protein. This is just to show you how massive these development efforts are. This is outdated. There's probably now more vaccine candidates, but here on the bottom, we have the clinical pipeline from preclinical to licensed. We have more than 140 vaccines in the preclinical phase, many in phase 1 and phase 2 trials. and by now it's 11 in phase 3 trials. So this is outdated. 2 are licensed, and there's an asterisk there because one of them is the Russian Gamaleya vaccine, which was licensed after phase 2, which is not what's usually done. The second one is a vaccine that has been developed in China by CanSino, and that was licensed, but only for use in the Chinese military. Now, a little bit of data from the non-human primate model, and don't worry, I'm not going through that whole slide. I would just want to extract the message here. So a lot of these vaccine candidates that are now in phase 3 have been tested in rhesus macaques or cynomolgus macaques. This includes the inactivated vaccines. This includes the viral vectors from AstraZeneca and Janssen, the Moderna RNA vaccine, and also the recombinant protein from Novavax. And the take-home message here is all of these vaccines protect non-human primates from disease. So basically, if you vaccinate these animals and you challenge them, you try to infect them, their lung is completely protected from a virus replication. But the other take-home message here is that in most of these experiments, the upper respiratory tract could still be infected, meaning there was still some replication, usually a little bit reduced, but still some replication in the upper respiratory tract. And that indicates that while these vaccines will protect from disease, they might not protect from getting an asymptomatic infection, and they might not be perfect in inhibiting transmission of the virus. So there might still be transmission from a vaccinated individual to another individual. So that's important to keep in mind. And then we, of course, also got phase 1 and phase 2 data for these vaccine candidates. I'm just going to give you explain the main message here. The inactivated vaccines were very tolerable. There were very few side effects in terms of reactogenicity. And they give intermediate neutralizing antibody titers. The vectored vaccines by CanSino and AstraZeneca, depending on what vaccine candidate it is, they can give okay neutralizing activity in serum or a very low neutralizing activity. This is the CanSino vector that's based on Ad5. And then we have the RNA vaccines, Moderna and Pfizer. And I have made the name of the Pfizer vaccine that actually made it to phase 3 bigger because they tested 2 initially. And I also highlighted here the doses that ended up in the phase 3 trials, because of course in phase 1 and 2 they tested different doses. So for Moderna, it's basically 2 shots with 100 microgram RNA dose. For Pfizer, it's 2 shots with a 30 microgram RNA dose. And what we see here, and I'm not going to go through all of this data, but these vaccines induce pretty good neutralizing antibody responses after the second vaccination. After the first vaccination, the response is not that great. And then we have Novavax, and this is a recombinant protein vaccine that's basically a recombinant version of the spike made in insect cells and adjuvanted. And it's the same here. If you give it once, the neutralizing activity in serum is not that great. However, if you give it twice, you get very, very high neutralizing titers in serum. So basically, all of these vaccines induce pretty good immune responses. And then this is just an overview of the vaccines in phase 3. We have the 2 RNA vaccines, Moderna and Pfizer. They have a check here because they actually finished phase 3 in terms of efficacy testing. And I'll show you the results. We have AstraZeneca, that's the Oxford vaccine. It's based on an adenovirus vector. We have interim results here for the efficacy. We have Janssen, which is a little bit behind. That's also a virus vector. And then we have Novavax. Novavax started phase 3 trials in the UK and in South Africa, but they didn't start trials yet in the US. They were planning to do it in November, but there were delays. Then we have the Gamaleya, the Russian vaccine candidate, for which we now also have interim results, which look pretty good. This is also based on viral vectors. We have 3 inactivated vaccines that are produced in China, but trialed globally. And then we have CanSino, which is an also an adenovirus vector vaccine that is also produced in China and trialed globally. And there was a recent new vaccine that has been also licensed in Russia based on phase 2 result, and that's called EpiVac Corona, but there's really not much data available for that one. So just a brief explanation how these trials work. I know most of you are very much familiar with that. Basically, you have two groups that are usually of equal size. 50% of them get the placebo, which in many cases is saline solution that's injected. In the case of AstraZeneca, it's another irrelevant licensed vaccine, and the other 50% get the vaccine. The studies are usually double-blinded. The vaccinee doesn't know if he or she is getting a placebo or the vaccine, and the person who's giving the vaccine also doesn't know. And then COVID-19 cases are reported in both groups. Usually for these trials, it's really COVID-19 cases, so symptomatic infections. They don't look at asymptomatic infections. And of course, the data is looked at by an independent data monitoring committee that has not much to do with, or is independent from the company that develops the vaccine, and is also independent from the medical centers that perform the trial. So for Pfizer, you all have heard that they had final results. They enrolled 43,000 individuals. They had 170 COVID cases in this group, 162 in the placebo group, 9 of which were severe, and 8 in the vaccine group, and 1 of the ones in the vaccine group was severe. They report a 95% efficacy. They started measuring a week after the second dose. And what's very important is they report a 94% efficacy in people 65 in the 65 to 85 year age range, which is important because that's really the high risk group, and very often vaccines don't work well in older individuals. They had no significant safety concerns, and as you know, they already filed an EUA. And the FDA is meeting to discuss this on December 10th. Pfizer said that they have already 50 million doses that they can provide in 2020, and they expect to have 1.3 billion doses in 2021 globally. And then yesterday we heard that Moderna also has their final results. They enrolled 30,000 individuals. They had 196 COVID cases, 33 of them in older individuals. 185 are in the placebo group. That included 30 severe cases and 1 death, and 11 in the vaccinated group. They had no severe cases in the vaccinated group. And they report a 94.1% efficacy, and that's measured 14 days after dose 2. They didn't do a sub-analysis, or at least didn't provide the data for the older individuals, which they also have in the study. They mentioned that there are no safety concerns, and they filed an EUA yesterday, and the FDA is meeting on December 17th to discuss that data. They expect to have 20 million doses for the US still in 2020, and they are planning to have 500 million to 1 billion doses ready 2021 globally. And then there's the AstraZeneca results, of which you probably have heard as well. They reported on 2 studies, and they started measuring efficacy 14 days after the second dose. So the viral vector was given twice. They had 62% efficacy in Brazil, which is based on a lot of people, and 90% efficacy in the UK, which is based on very few people for a phase 3 trial. And they said that they had 131 COVID-19 cases, but they didn't tell us the distribution. And then it turned out that the UK group got only a half dose as first shot due to an error, a calculation error. And so I think here the data situation is a little bit unclear. I think the only thing that we can say right now is that the vaccine works, but we cannot say how well it works. And of course, having to admit that there was a dosing error does not increase public trust and confidence in that vaccine candidate. And that's something that you wouldn't expect from a major vaccine company, actually. So just a few words about reactogenicity and safety. A lot of these vaccines have relatively strong reactogenicity. Meaning injection site pain and flu-like symptoms, including headache, increased temperature, and fatigue, which in most cases is transient, but it's more severe than or more frequent than you would have it from a regular flu shot, for example. And the reactogenicity is highest with the adenovirus vaccines, followed by the mRNA vaccines, followed by the recombinant and inactivated vaccines, which have a pretty good reactogenicity profile. And I wanted to show this here on the right side. These are data from Moderna. Remember, they used the 100 microgram dose in the end, and you have reactogenicity after the first shot and after the second shot. And what you can clearly see here is that it gets worse after the second shot. So very few people have systemic reactogenicity after the first shot. Most of them have it after the second shot. That's something to keep in mind. And because children usually have worse reaction in terms of reactogenicity, it might be a problem to license some of these vaccines in children, specifically in small children. The other point that I wanted to make is immune responses in older people. Although Pfizer already addressed it and they see good efficacy, it's worth pointing out that older individuals, and this is shown here on the right side, please focus on the blue bars, which are the vaccine candidate that moved forward, seem to have lower neutralizing activity after vaccination than younger individuals. This might or might not become a problem. It seems in the phase 3 trials, it did not impact on efficacy. It might be that you need very, very low neutralizing titers to be protected. Since we don't know the correlate of protection yet, we can't really tell yet. And then a few other issues that I wanted to highlight. One is the production, specifically large-scale production. Some of these technologies are new and have never been produced for the market. Some of them are produced by companies that don't have vaccines on the market, like Moderna or Novavax. And so we have to be careful. There might be hiccups with supply chains, with how many vaccines they think they can make and they can make in the end. So that's something to keep an eye on. Distribution is in a way a challenge, specifically for the RNA vaccines. The Moderna vaccine needs to be stored at minus 20 degrees Celsius. The Pfizer vaccine is at minus 80 degrees Celsius. And this is doable, but it's a challenge in high-income countries. And it's probably really a big challenge and maybe impossible in low-income countries. So that's something we have to keep an eye on. The administration is also a question. First of all, if you have a lot of vaccine and we want to vaccinate a lot of people, how are we going to do that? And then specifically initially, when there's little vaccine available, how are we going to prioritize? And there's by now pretty good plans for that, for vaccinating healthcare workers and high-risk groups first, but those need to be implemented and followed. And the last point is immunity. You know, the situation won't change overnight just because we have a vaccine. It takes some time until you are protected, right? So the first shot doesn't do that much, specifically with the RNA vaccines, and you really need the second shot. And after the second shot, it still takes about 2 weeks until your peak antibody titers are reached. So it's depending on the vaccination regimen, 5 to 6 weeks from the first vaccination until you are protected from disease. And so to conclude about the vaccines, there are several candidates that induce strong neutralizing antibody responses in non-human primates and in humans. This is great news, and I hope a lot of them make it through phase 3, because one company or two companies cannot really produce vaccines for the global market, for the global demand. So we need a lot of vaccine companies that can make these vaccines. The protection from lower respiratory tract infection in the non-human primate model is really solid, and that's basically protection from disease. While there was less protection of the upper respiratory tract. And that's often a problem with injected vaccines because they mostly induce a systemic immune response, but not really a local mucosal immune response. And so we have to look at that in phase 4 trials and probably in additional clinical trials to see how much transmission and how much infection in general, asymptomatic infection, is actually impacted by these vaccine candidates. I don't see that as a problem right now. We need vaccines to mitigate the impact of the pandemic, But in the long run, this might be important to prevent outbreaks, in cohorts where, for example, healthcare workers might get asymptomatically infected. And they might then interact with vulnerable patients that cannot get vaccinated, and they might infect them. So this is certainly something we have to keep an eye on. The Pfizer and Moderna phase 3 trial results look really good. Again, we're in the 94-95% efficacy range. That's almost as good as it gets for vaccines. Of course, measles is better than that, but that's almost an exception. And the other vaccine candidates look pretty good too. It seems that it's relatively easy, and knock on wood, to make vaccines against this SARS coronavirus too. And with that, I just wanted to acknowledge all my collaborators and my team here. Not going through everybody here, but since the pandemic started, we started so many collaborations within Mount Sinai. Everybody stepped up, everybody worked together. We have now very nice collaborations with the pathology department, with a lot of people in the hospital, and of course within the microbiology department, and also with people from other institutions. And of course, I also wanted to thank my funding sources. Our funding is mostly coming from NIAID, from the civic centers, and from SEERS. And with that, I'll conclude, and I'm happy to take any questions.
Great, thank you, Dr. Krammer, that was terrific. So I see we already have some questions in the chat. Florian, is it helpful for me to read them, or do you want to read them? Do you see them?
Yeah, that would be great.
Sure. So first question is, how long the T memory cells last, and what is their lifetime, and can you explain the significance CD4 immunity?
So there is data now out to 8 months, and so all kinds of memory cells were stable over that period of time. There's a very nice preprint out from Shane Crowley's group from La Jolla Institute of Immunology. There's another one from Rockefeller, and another one from a group in Seattle. What the significance of the different cellular responses is unclear. Of course, if you want a good antibody response, you also need a good CD4 response. But to what degree there is correlation between protection and different T cell responses is not clear yet. We see first correlations between antibody responses and protection, which is great. And antibodies are easy to measure, so that's easier to do. And I think we'll have to wait a little bit longer for data of correlation between different cellular immune responses and protection. I'm pretty sure they're there for some of the subsets, but it's just harder to study that.
There's a question here for one of our people about all the patients asking about the long-term safety and efficacy of the mRNA vaccine, and is it true that these are the first mRNA vaccines to be used?
So they're the first mRNA vaccines to be licensed or to be authorized to be used at large scale. The first mRNA vaccine clinical trial started in 2013. There have been numerous RNA vaccines that have been tested in humans in phase 1 and phase 2 trials against infectious disease, but also as tumor vaccines or cancer vaccines. So hundreds of people have been vaccinated with those since 2013. And there were no real safety signal in terms of long-term safety. It's true that, these trials, the phase 3 trials started in July. And so there's data in the phase 3 trial, at least from July on. But you also have to keep in mind that a lot of the things that, are rare, but can occur as side effects of vaccines that are more serious, like Guillain-Barré syndrome or narcolepsy, actually occur relatively quickly after vaccination. They don't occur after 15 years. And typically phase 3 trials also don't last 15 years. They last 2 to 3 years. So because there are so many people in these trials, there's not that much concern about safety, honestly. At least that's how I see it.
Thanks, Florian. There's a question, which is more important for protection, antibody titer or T-cell response?
Well, you're asking an antibody person. I'm going to tell you antibody titer.
Okay. How about this? Are there side effects from the mRNA vaccine other than injection site flu-like symptoms that you may anticipate?
So not really. They didn't report any kind of problematic side effects. In some studies, they saw diarrhea in a small percentage of individuals. But again, I think it's important to stress that the reactogenicity, the flu-like symptoms, and the injection site pain can be much more, or is much more frequent, and in some cases much more severe than what you would see from other licensed vaccines, right? So, you know, I think if we vaccinate people, we need to really make clear that that might happen, that they might feel sick for 24 or 48 hours. I think that's really important to point out, because if that's not communicated, we will start to vaccinate tens of thousands, hundreds of thousands of people, and all of them will be back or will be calling the next day and telling us that the vaccine made them sick.
Gotcha. It looks like safety is definitely on people's mind. And there's another question it's asking, is it really accurate to claim there has been no compromise on safety when the EUA applications will only contain 2 months safety data for 50% of participants? Don't we usually have much more safety data before vaccines are marketed?
It really depends on the candidates, right? For some of these phase 3 trials, you have relatively few individuals, and you follow them for a longer time, right? Which, if you have fewer individuals, you also need to do that because you need to have enough events, enough people getting infected, right? In order to determine if there is efficacy. So I think from my point of view, we have enough safety data. And again, you have to keep in mind the situation, and that's what these EUAs are for. We are on one, it's kind of a balancing risk, right? We have now 40,000 people who have received these vaccines, and I'm basically adding up the people who got the vaccine in the Pfizer trial and the people who got the vaccine in the Moderna trial, and leaving out the placebos. And we don't have any safety concerns with those 40,000 people, plus the historic RNA trials. On the other side, we have a virus that, you know, out of 1,000 people kills between 3 and 15, sends a lot of other people to the hospital, and causes long-term issues. So I think you have to balance the risk, and if you do that risk analysis, I think it's clear that for this situation, the vaccine is pretty safe.
Gotcha. Florian, there's a question if you could comment on the Janssen vaccine efficacy as compared to Pfizer.
Janssen did not report any efficacy data yet. So they started the phase 3 trial. They showed pretty good data in non-human primates. Their vaccine is only given once, and we'll see how that works out. So that's not clear yet.
Gotcha. Is there a reason to believe that people who have had organic exposure to COVID-19 would not require vaccination? So
protection is, and there's some data that indicates that already, protection is always a question of how much, right? And so it's really hard to say without a correlate of protection, how much, if somebody is protected or not based on their antibody titer, right? Again, we know now that more is better, but we don't know how much you need to be protected. And so I think it's not a bad idea for people who had exposure to SARS coronavirus 2 and who may have antibodies to still get the vaccine. And the second point here is that it might also be challenging to test everybody before they get vaccinated. Because of course we are talking about 330 million Americans. And so I think it would make sense to also vaccinate people who already have immunity or already have antibodies.
And there's a corollary question here that's related, Florian. It says a small number of people seem to have had a second clinical infection. Is it known what type of response they had to the first infection or why it was not protective?
Yes and no. So we know, for example, from the case in Hong Kong that person didn't make good antibodies after the first exposure and then had a mild infection after this, or an asymptomatic infection for the second time and then mounted an antibody response. There's a second case in Utah where we don't know how the antibody response looked after the first time. We know that that person responded with an IgM response the second time, which might indicate that maybe there were no antibodies after the first time. And then you, of course, also have cases of reinfection in people who have who are immune suppressed. But in general, you know, these are not absolute things. You cannot say somebody who was infected already can never be infected again. We know that reinfections can occur with human coronaviruses in some intervals. So it's probably a question of probability, right? And the question of how strong your immune response is. But in general, what studies start to see is that you certainly get a degree of protection from a primary infection.
So here's a comment and a question. Congratulations to you on your beautiful studies and for sharing the information about your assay with the entire scientific community! Exclamation point. Thank you, Dr. Coleman. Do you think that there will be safety concerns if people are vaccinated with more than one vaccine?
Yeah, that's a very good question. And especially if there is 3, 4 different vaccines with different platforms out there, this might happen, right? Because people might just get a vaccine and then they might get a second vaccine and it's hard to control. I don't think that there are necessarily safety concerns about that. We know from other pathogens like influenza that giving one type of vaccine followed by another type of vaccine sometimes can give you a superior immune response. And I suspect that there will be a lot of academic trials that actually will start to look into that. I don't see an immediate safety concern if somebody gets 2 different vaccines.
Gotcha. Florian, someone asking a question about a timeline for a vaccine development for children, or if you could comment on the children getting vaccines.
So I'm not 100% sure if they already started to age de-escalate or if it's just planned. But what you typically do is you start in basically older children, or people in the 16 to 18 year range. You start to use you start a trial testing the vaccines there, and then you go down by age, right? And so this will be done. The question really is in which age group will the vaccine be tolerable? In which age group will we have reactogenicity that's okay? And in which age group will there be a problem, right? And the trials will show that. But I don't think that that children will get vaccinated right away, and it might take some time until we have something that can be used in children. I'm very hopeful about the recombinant protein vaccines just because they have a very nice tolerability profile. And they might be a good choice for children, but we'll see. I mean, we need to get the data in order to kind of be able to make these conclusions.
Gotcha. And what do we know about vaccination in immunocompromised patients?
Well, not too much. Of course, the clinical trials probably had a proportion of people who were immunocompromised to some extent. I don't know the exact inclusion and exclusion criteria. But of course, that's always an issue. If somebody is immunocompromised, it depends on the degree of and type of problem with the immune system. The immune response might be suboptimal, right? And that might lead to suboptimal protection. I think it would still make sense to vaccinate people who are immunocompromised. But I think, again, we will need data on that moving forward.
Gotcha. And staying on that theme, which antibody test would you recommend if someone wants to check to see if they have a good immune response 2 weeks after the second dose?
Of course, our antibody, that's no question.
And then coming up on the end, there's a lot of questions. If I miss some of them, I apologize as they keep popping in. If the virus mutates as it has, will it matter as far as vaccine protection?
That's a very good question. Viruses mutate all the time, right? Specifically RNA viruses. There are two things that you have to keep in mind about it. The first one is we all know about influenza, antigenic drift, revaccination. We have to change the vaccine all the time. That's an exception. The measles vaccine was developed in the 60s. It's an RNA virus and it mutated. It mutates quite a bit. The vaccine still works very well. So flu is really an exception here. And in addition to that, coronaviruses, which is very rare for viruses or very unusual, have a proofreading activity. So they make fewer errors than other RNA viruses. But of course, there have been mutations found in virus that was circulating in mink in Denmark, in the Netherlands. We have a cluster 5 in Europe that has a mutation in the RBD. And so the first data that's out says that those might not be drastic changes that they might not impact on vaccine efficacy. But of course, we need to monitor that over time. But in general, coronaviruses don't drift the way influenza virus does. Influenza is really a different beast.
Gotcha. And one last question. Why did Moderna and Pfizer use an RNA vaccine model rather than one with less reactogenicity or more historical use?
Well, for Moderna, there's an easy explanation. Moderna is an RNA vaccine company. That's what they do. For Pfizer, it might have just been the speed with which they could respond, right? And they partnered with a small German biotech company that's actually called BioNTech. And they are experts in RNA vaccine development. And I think that's the reason why they went for that, because they could do this quickly. There were, of course, other vaccine companies that, like Novavax, that use recombinant proteins, like Janssen, that use adenovirus. So there were different choices made, and the RNA technology is very promising. I know it's relatively new, but if you look at the results that you get in some of the clinical trials and also in animal models, it's just phenomenal immune responses. It was clear that the reactogenicity might be an issue, but it's not as bad as I might have explained. I'm just trying to warn people that they should expect some of that. But as I said, I mean, for Moderna, it was a clear pathway forward, and Pfizer chose that pathway because they could do it quickly.
Thank you, Dr. Krammer. Well, listen, we had a huge turnout today with over 200 people, which was just wonderful. And it's great to have you as such an expert in this area, at our own institution. It was just terrific, Florian, your presentation. And I know I speak for everybody on the call. Thank you very much, and for all the great work that you're doing for us at Sinai. So I'm sure as everyone walks by you in a mask, say hi to Florian and congratulate him on his great work. So have a great day, everybody. Florian, thanks a lot.
Thanks for having me.
Yeah, absolutely.